Okay, we get to talk about chapter 6 and infection. So, to start off, we need to talk about some microorganisms. So what are microorganisms? These are small living forms. They include bacteria, fungi, protozoa, and viruses. And many can grow in an artificial culture medium. They are non-pathogenic, which means that they usually don't cause a disease unless the conditions of their host, their environment, etc., mutations exchange. They're part of the normal flora, and often these are beneficial to us. There are also pathogenic ones, and these are disease-causing microbes. So with the different types of microbes, this is figure 6.1 in your book on page 89. This is talking about the different shapes. Okay, so you have cocci, which are small little circles, bacillus, which are kind of rods, vibrio, which are kind of squiggles, spirochetes, spirilla, right? Those are like the spiral spaghetti noodles. Pleomorphic, streptococci, or strepto meaning lots of them. So you have a lot of the different microorganisms that you can, the shapes and sizes that they come in. Then down here in B, this is going to be what a virus looks like. And it has a glycoprotein cover, then it has an envelope, it has the capsimer, which is in the middle, and then the nucleic acid capsid, the core protein, and all of those together are the nucleocapsid. So the fun thing about viruses are they can't reproduce without taking over a host organism. And so, we're in the middle of a viral pandemic right now with the COVID-19 virus. So you guys probably know more about viruses than you ever wanted to know. And viruses are one of the two things that really scare me because they mutate so incredibly easily, and I mean, they're hijackers and they literally take over the host cell's body to hijack their cells and say, nope, you're making my DNA instead of, the host body that you're in. And then over here in C, you have an example of a fungus. And this is where you would have like a blastosphere or a chlamydia spore. Okay, so those are different types of microorganisms. So when we're talking specifically about bacteria, these are classified as prokaryotic cells. Prokaryotes mean that they're like pro or before eukaryotic cells. So they're the simple cells. And so because of that, they have no nuclear membrane. Therefore, they have no nucleus. So all of their genetic information, their cellular information just kind of floats freely around in the cell itself, where we have eukaryotic cells, which are DNA and that stuff is contained within a nucleus. But the prokaryotic cells, those are like the primordial or the more simple cells, and so they don't have that organizational level. Um, they function metabolically and they are able to produce. They divide by binary fission. They have a complex cell wall structure, and they do not require living tissues to survive. They also vary in size and shape. So there are a couple different major groups of bacteria. You have your bacilli, and these are going to be your rod-shaped organisms. You have a spirochete, which they kind of remind me of spiral macaroni. Sorry, guys, I probably just ruined macaroni and cheese for you guys. But these include the spiral forms and vibrio. And then you have your cocci, and those are like the little like round guys, and then they are the spherical forms. You have a diplococci, streptococci, or staphylococci, right? So this would be like the ones that cause strep throat or staph infections, right? Um, the structure of a bacterium and the mode of action of the antimicrobial drugs are shown in this figure. And this is figure 6.3 on page 91 of your book. And this is titled The Structure of a Bacterium and the Mode of Action of Antibacterial Drugs. So up here in step 1, it says that there's a bacterial cell wall, and there's defective synthesis, for example, with penicillin bactericidal. So it causes this little hole, right? You can also have metabolic interference, and this would be through the action of sulfonamides. You have over here in 3, it shows that you have an interference with the chromosome, DNA, or protein synthesis, and this would be like through the actions of tetracyclines or bacteriostatic antimicrobial drugs. And then down here in 4, it shows that there are some antimicrobial drugs that can cause cell membrane damage, and an example of that would be a polymyxin. Okay, so you can see that we can target different parts of the bacterial cell in order to make effective antimicrobial drugs. So talking about the basic structure again of the bacteria, we have some toxins. We have exotoxins, endotoxins, and enzymes. So again, look for those root words. So exotoxins, these are going to be usually produced by gram-positive bacteria, and they are secreted or excreted, right, from the organism. So the exo going out. Where the endotoxins, these are going to be present in the cell wall of gram-negative bacteria, and they're released upon the death of the bacteria. And these are vasoactive components that can cause the septic shock. So again, look for those keywords. Vaso is the vascular system, right? So your blood vessels. And so these, obviously, we just got done talking about septic shock in the last couple units, right? And so if you have some of these toxins that are released, it can cause, spasming and problems, vasoconstriction, lots of problems with your blood vessels, and it can cause or start to lead to septic shock. Then you also can have enzymes, and these are going to damage tissues and also promote the spread of infection. So what about spores? Spores, they're actually kind of cool. I mean, they're also kind of scary because they're really resilient, you guys, right? But they're very preservative, right? And so they help preserve that object for a long time. So spores are formed by several different species. They have a dormant latent form of the bacterium, and that just means the spore can be hanging out for a long time, and then when it gets exposed to the right environment, and this could be decades later, then boop, it just kind of pops up again and it starts growing. And so because of this, it can survive long periods of time in the spore state. And they're also highly resistant to heat and disinfectants. And so there was some story, and I need to kind of figure it out and which one it was. But maybe I'll try to do that before lecture. But there was a story that I read a long time ago, and it was somebody was going through like an old canning room, bomb shelter, like underground storage something, and they found like a canning jar of something, and they opened it up and it re-released spores that it was like a really bad thing, and it was kind of like a disease that was eradicated in the present time, but since that person re-released them, it started to contaminate and it became a problem again. I'll have to look that up because it was an interesting story. So what about our viruses? Well, like I said, these guys kind of scare me. I'm looking at thrush in your fun thing. Stop and look at your picture and figure 6.9 picture B on page 97 and thank me later. Picture of oral thrush. It happens, guys. It's just, it is what it is. All right, so anyway, viruses. These guys, again, they're kind of scary to me because they mutate so incredibly rapid, and also they're hijackers, right? Like I said a couple slides ago. So these are small obligate intercellular parasites. So obligate just means that they're dependent on something else in order to grow, right? So they can't do it by themselves, and they're within the cell, right? Because it takes over the nuclear area. Parasitic because, again, they can't do anything without hijacking and taking over a host organism. And so this is a very good, scary summary sentence. They also have a protein coat or a capsid around them, which makes them very resilient. The protein coat comes in various shapes and sizes, and again, it can mutate or change very quickly and adapt. We're 1 year into COVID, and we already have, I know, at least 3 known variants, but it might be up to like 5 now, right, of them. And unfortunately, the variants are becoming even more dangerous than the original COVID-19 was. The nucleic acid is either DNA or RNA. The classification dependent on the nucleic acid that's present, and some RNA-containing viruses contain reverse transcriptase enzyme, and that can convert RNA to DNA. So again, these are kind of like little alien cells. Again, they terrify me. Okay, I'm sure I've told you that like multiple times now, and you're probably laughing at me and saying, "Oh my God, my teacher's so crazy," but they are very scary to me because they mutate. They're quick, they're smart for a little non-living thing, right? So there's debate on whether they're living or non-living, and one of the definitions of being able to be considered life is being able to reproduce like by yourself, like with your own kind, right, by yourself. And since viruses can't do that, they're technically not living, but man, they're incredibly smart, and they just mutate and just hijack and take over things so incredibly quickly. Um, so what about the different shapes of viruses? Well, um, you have the circles. Hold on a second, I'm trying to find this picture. And yo, checked up, and it's I just went right past it and didn't even see it. So, but the various shapes you have, again, you're going to have that envelope, right? You have the protein coat, that's the capsid. You have the DNA or RNA that's going to be in the middle, right? So this is basically on figure 6.7 on page 95. And so these are just the different shapes of the virus. Okay, so you can have a circle, you can have like a that would be like a hexagon, pentagon, right? In an envelope. You can have the alien looking ones. These are the normal viruses that we see, and then this is what COVID looks like with the spikies around the outside of it. Okay. So when you have an active viral infection, the virus is going to attach to the host cell. The viral genetic material is going to then enter the cell, and then it takes control of the cell. Like I said, it's a hijacker, and it uses the host cell to synthesize. And remember, synthesize is just a fancy word for make. So it's going to make the viral proteins and nucleic acids. So again, it's kind of hijacking and say you're no longer making stuff for your human body, you're making the things that I'm telling you to as a virus. And new viruses are assembled in the cytoplasm of the cell, and then the viruses are released by lysis, which remember is splitting, right, of the host cell, or by budding from a host cell membrane. So if you think about this, okay, not only does the virus come in, it invades, it takes over the DNA replication system, but then at the end, it explodes the cell, guys, to be like, spread little viral babies everywhere. Or if it's nice, it can bud off, right? But a lot of times it causes this huge lysis or explosion of the actual host cell. And so then, the cell that didn't have a choice anyway, but was being nice, right? Not that cells really have minds, but whatever, we'll pretend. And like made all this stuff because it was hijacked and was told to, then it gets killed at the end. And it's like, come on, Mr. Virus, you're being really mean. And this is shown really well in the diagram that's under the one that I just showed you on figure 6.7 on page 95. Okay. I think I skipped one. Okay, and then yeah, no, we're good. And then the virus is going to enter the cell with an active infection, and then the viral proteins can be produced and inserted into the membrane of the host cell, and then this may stimulate an immune response and therefore destroy the host cell. And then the viruses may reproduce actively if the immune system is depressed. An example, this would be like the herpes virus, right? So it just kind of takes over and it suppresses your immune system, and so you can be prone to getting even more infections and stuff because your immune system is repressed. So when you're talking about the principles of infection, the infection is going to be where the organism is able to reproduce in or on the body's tissue. If it's sporadic, then this is going to be occurring in a single individual, where if it's endemic, then it continues transmission within a population. If it's an epidemic, then it's higher than normal transmissions or is spread to a new geographical area. And then again, if we think of COVID-19, we're in the middle of a pandemic, and this is where the transmission has occurred on most of the continents, right? So it started local in Wuhan, China, and then it started spreading to different countries, and then it came here, and it's just keeps spreading. So for transmission, how do things happen to be transmitted? So they can be transmitted from person to person. And I realize as I'm licking my fingers, actually has a funny story. I think I already told you guys this one. So it was like last fall, winter. It was really tragic. My neighbor, actually, you guys weren't in my class. So my neighbor's husband passed away. He was a very nice man, very great guy. And so I did the nice neighbor thing and I made him a pan of lasagna. So I'm like a home canner, right? So like, home canned tomatoes tend to be a little bit soupier, a little bit waterier, whatever. So I made sauce and this whole huge pan of lasagna. Now remember, guys, we're in the middle of a pandemic, and yours brainiac professor here, who does have her PhD in neuroscience, right, walks over and hands this pan of lasagna to my neighbor's son, and I got like juice, the tomato juice all over me because I had made it really big. Stupid me, not thinking. He went to go get me a towel. I'm like, oh no, I'm good. Totally licked it off my hand and then wiped it on my pants. Now, I'm usually not that much of a slob either, but it was just one of those like, oh no, I got it, no worries, don't like worry. Then I like left and I was like mortified because I'd only met like the kids like one other time, and here I'm giving them food and I'm like licking my hand in the middle of a freaking pandemic. So I'm sure they probably tossed the lasagna. I probably would have too, or boiled it for extra long and then or boiled, cooked, whatever. But sometimes I'm not so smart. Okay, anyway, so transmission of it is going to be from person to person. If there's a reservoir, then that would be the source of the infection, the person with an active infection, or a person who is asymptomatic. And then there's a carrier, and that would be a person that may never develop the disease but still is a carrier, and a person with a subclinical signs of the disease. So right now we're in the middle of this with, again, COVID. My sister is a good example of this, right? She did an antibody test for COVID and she tested positive for antibodies, but never ever exhibited any of the signs and symptoms. So at some time she was a carrier, and she's actually very careful. So I think it was a false positive on the antibody test, but I'm just a scientist, so what do I know? All right, so how can we transmit the infectious agents? Well, there are a couple different links in the infection chain. One is that we have an agent, and this is where the microbe is going to cause the infection. And then the reservoir is going to be the environmental source, such as contaminated soil or an infected person or an animal. The person can carry the agent and show no signs or symptoms, or the person or animal may show signs and symptoms of the disease. So this is kind of like when it came over from Wuhan, right? We had people that flew on airplanes that were asymptomatic, right? Or they might have had sniffles, but I mean, we've all flown with colds before, so it's like not the biggest of deals until we're in the middle of a pandemic, and now it's a huge deal, right? But yeah, just things happen. Um, we also have the portal of exit, and this means whereby the agent leaves the reservoir. Then you also have a mode of transmission, and this is the method where the agent reaches a new susceptible host. So air, water, direct contact, food. Again, with COVID, if I sneeze, it's a aerosolized germs, right? And then the next person can walk into my little germ pot of sneeze and they can get it. Also, sharing water glasses with my husband or something like that, direct contact, giving my husband a smooch or something that would, obviously transmit it. And then, food could also be, ill prepared. I think that the original one, I think they tracked it down to a wet market in Wuhan, China, where it was one of the workers, I think, was sick and killed a bat or a rat or something. I don't know. There's still speculation about the difference between how it started. Okay, and then the portal of entry is how it accesses the new host. And the susceptible host, the susceptibility will depend on the health status of the individual, their level of immunity, their age, and also their nutritional level. So when we look at this diagram, this is the transmission of the infectious agents. And so this is on page 101 of your book, and this is figure 6.13. And so this just kind of shows different ways that they can be spread. So you have the source here, where you can get it excreted by the oral or nasal secretions. Then that can get contact with utensils, your hands, or your lungs, right? You can also then, touch. I know I keep sniffling and touching my hands and my mouth and stuff like that. I just washed my hands before I started to do this lecture. Still shouldn't do it, but my nose is running for some reason. But anyway, we can inhale it. We can have those utensils then touch our lips. We can have contaminated eggs, like think of salmonella, right? Eggs, chicken, or water that can again touch our lips. So uncooked, undercooked chicken, raw eggs, utensils that weren't cleaned, people that aren't washing their hands after they go to the restroom, etc. Swimming in stagnant water or infected water that was contaminated. We all do this growing up. We put water in our mouth and spit it out, right? And it's kind of fun. Then also you can have injections that are going to puncture, the skin and potentially have direct access to the blood with, drug people. I think they inject heroin right and things right into their blood. So not my realm of knowledge. Also, injury can cause broken skin, etc. Bee stings, ticks, worms, leeches, right? Things like that. They can break the skin. You can also have animals. They can be carriers. So this is the thing why you're not supposed to like run around barefoot in places where dogs are or pigs or chickens because you can, get scrapes on your feet and you can pick up things. Also, sexual intercourse. Just direct transmission of the fluids, but also you can have microabrasions when you're having sex. And so then you can have, transmission through that through just small little tears in the penile coating or in your vaginal wall. Um, so you can have direct contact, which would mean that there's no intermediary. There's no middleman, right? It's just direct contact. And so this would be an example of like touching the infectious lesion, sexual activity, contact with the infected blood or bodily secretions. Or you can have some indirect contact, which involves intermediate objects or organisms, contaminated hands or feet, food, right? Or you can have fomites, which are inanimate objects, door handles, glasses, you know, like the debit card things, right? Anything that is not a living thing, but it can have contact from people before and after you. You can also have droplet transmission, and this would be respiratory or salivary excretions that are going to be expelled from the infected individual. Aerosolized transmission, which involves small particles from the respiratory tract, and these would be suspended in the air and they can travel farther than the droplets. You can also have vector-borne transmission. This would be with an insect or an animal as an intermediate source. When we talk about nosocomial infections, these occur in healthcare facilities, such as hospitals, nursing homes, physician offices, and dental offices. And about 10 to 15% of patients acquire an infection in a hospital because there's a lot of bugs that are present, right? People go there because they're sick, not to hang out and get like a pedicure, right? So there are many microbes that are present. And then there are patients, again, that may be there for something completely unrelated, but they might have an undiagnosed infectious disease. You have a shared environment, which means shared air, even though it's getting filtered and the air exchange is amazing, you still have problems, right? You can also have treatment that may cause weakened immune systems. And usually when people are sick, their immune system isn't like, Johnny on the spot. And then also many healthcare workers and fomites act as reservoirs, elevators, door handles, even now like the hand sanitizer things or, leaving the bathroom and people don't wash their hands. I don't know why, but they don't, whatever, things like that. Um, so when you're looking at host resistance and microbial virulence, this is box 6.1, and this is shown on page 101. And this shows the host resistance and then also increased microbial virulence. So for instance, the intact skin and mucous membrane, the body secretions that we just talked about, non-specific phagocytosis, all of those things. And then it can have production of exotoxins and endotoxins, and then destructive enzymes, spore formation, etc. So what about factors that decrease host resistance? Well, this would be age. So infants and older adults, they have the weakened immune systems, right? Babies that haven't been around enough or long enough to like actually gain a strong, robust immune system. Then older adults aren't as out and about as, middle-aged and younger people are. Pregnancy, genetic susceptibility, immunodeficiency, malnutrition, chronic disease, severe physical or emotional stress, as well as inflammation or trauma. You can also have impaired inflammatory responses that can decrease your host resistance. So what about virulence and pathogenicity? Well, what is pathogenicity? This is the capability of a microbe to cause an actual disease. And then the virulence is the degree of pathogenicity. So examples of that would be invasive qualities. For instance, how well they can move, or if they're enzymes, if they're toxins, if they can stick to tissue by the pili, the fimbriae, the specific receptor sites. So like the little appendages, right? So a flagella, the pili are the little hairy guys. The fimbriae are again the little squiggly, taily kind of guys, and that can help adhere or stick to tissue. And then the avoid host defenses as well. Um, so, we are in the middle of a pandemic. These slides are from before COVID was a thing, because they came from the book from, 2019, so a hot minute ago. But we do have newly emerging diseases. We're in the middle of a pandemic. Let's see, we can, I shouldn't say this on camera. Turn this into a drinking game. How many times does Janet say that we're in the middle of a pandemic, right? Drinking water is what I'm saying, right? But there are different strains of common infections, such as the flu. I mean, people get flu shots. The flu shot is 50-50 guess on what strains are going to be virulent that year, and again, it's a virus and it continuously is mutating. It also can spread beyond the normal endemic areas and make it more into a pandemic like we are in the middle of. We also can have superinfections, and some examples of this would be multi-drug resistant forms of existing diseases such as tuberculosis or Staph aureus, so staph infections, right? So these are those superbugs that, get resistant to antibiotics because, again, they mutate or, the strongest survive. People don't finish their prescribed routine of antibiotics, and so then the microbes that end up living are the ones that are stronger that were able to, withstand and live through, the course of antibiotics, etc. So, how do we control transmission and infection? Well, infection control requires two approaches. One is the standard precautions that are used in all settings with clients when body fluids may be exchanged. And another one is specific precautions in clients that are diagnosed with a particular infection. And these are used in addition to standard precautions. So that would be potentially like roping off an area you're in quarantine. If you ever watched I Love Code Black, right? And they had a thing where they had to, you know, wall off parts of the hospital and things like that to try to, keep them separated so that it didn't spread because they didn't know what it was. All right, so what about the infection cycle and breaking the chain? Hi, Sophie. Guys, I swear to God, I'm like normal. But my cat, she sneezed, and so I looked at her, and one eye was down and one eye was open, like she was winking at me for like the longest time. She just sneezed again. She's really cute. Okay, this is on page 103. And this is the infection cycle and how we can break the chain. And so basically, if you have a susceptible host down here, well, first of all, we start over here with the entry, and then you can stop that by having current immunizations, right? But if you don't, then you have a susceptible host, and then depending on the health status, the point of entry, and the number of microbes, they can either become a carrier and then a reservoir, or they can have an incubation period where they're asymptomatic and then they can have the infectious disease. Then we can break it here, or they can become a reservoir, and then the source would be like a person, animal, insect, environmental area in the home, workplace, or community, such as the bus or the store, right? And then we can also reduce the numbers of like mask wearing and stuff so that things can't exit through our mouth, skin, and intestine, and then those barriers to, again, stop the transmission. So this would be where the masks would be, where you could break that chain. And then the transmission can be either direct or indirect. And the direct would be saliva, blood, poop, semen, exudates from the skin. And you can stop that with barriers such as gloves, glasses, gowns, and safe waste disposal. The indirect contact can be respiratory droplets on hands and surfaces, insect bites, which are vectors, and contaminated food and water. And then this can lead to hand washing, where you can disinfect, sterilize, do correct food handling and cooking, as well as water treatment to break that cycle so that we don't have entry into our mouth, broken skin, vagina, etc. So you can see that there's multiple different ways that hygiene is a big one, and get your vaccinations and just be a good person and don't sneeze on people, and that would be good too. So when we go to break the cycle, we have to locate and remove or isolate the reservoir of the infection. Then we're going to identify and restrict the access to the contaminated food or water, reduce the contact between the infected persons and the remainder of the populations, block portals of exit and entry, remove or block modes of transmission, and also reduce host susceptibility by immunizations, adequate nutrition, and access to healthcare. Additional techniques to reduce transmission would be adequate cleaning of the surroundings and clothing, sterilization, disinfectants, and antiseptics. So what about the actual physiology of infection? So this is on page 105 in your book. You have the incubation period, and this is the time between the entry of the organism into the body and also the appearance of clinical signs of the disease. And this varies considerably with the different organisms and what's incubating or being transmitted. You have the prodromal period. This is where you have fatigue, loss of appetite, headache, some non-specific feelings like, I don't know, I just don't feel right, but I know I'm coming down with something. And this is more evident in some infections than in others. Then you also have the acute period where we have infectious diseases that will fully develop. Um, how do we disinfect? Well, you can sterilize equipment by chemicals, heat in an autoclave, on other parts. And so a nice little side note is equipment must be cleaned prior to the sterilization or it will remain contaminated. So this is a very important thing to point out as well. Also use proper chemicals where antiseptics are going to be used on the skin and living tissues, where disinfectants are used on inanimate surfaces or objects. So with the patterns of infection, you have local infections, focal infections, systemic infections such as septicemia, bacteremia, toxemia, viremia. You have mixed infections, you have primary infections, and you have secondary infections or subclinical infections. And this is a nice little diagram. It's figure 6.15 in your book on page 105. And this is causes and development. So it's titled Onset and Possible Courses of the Infection. So down here in one, it says the pathogen enters the host, then the pathogen is going to colonize the appropriate site, and then the pathogen reproduces rapidly. Then you have the prodromal signs that may appear, and then the acute signs that are present. And then over here in 6, you have decreased reproduction and death of the pathogens. That's what we're trying to do, right? So the host defenses take effect, the nutrient supply decreases, the waste and the cell debris increases, and also you can take an antibacterial drug. And then you would have recovery that would occur down here. If it doesn't completely go away, it can go into a chronic infection where you have mild signs that are destructive but not as lethal, or you can have total recovery. Or if that doesn't work or you don't get treatment and you have an overwhelming infection, then this can cause septicemia and therefore death. So with signs and symptoms of infection, the local signs of inflammation would be pain, swelling, redness, and warmth. If it's bacterial, you can have a purulent exudate. If it's viral, then you have a serious clear exudate. So purulent is like the pussy kind of creamy, gross stuff. Where if it's viral, it's going to be like clear, almost like a serum in a way. Systemic signs of inflammation include that a fever may be present. You can have fatigue and weakness. You can have headache, and you can also have nausea. So how do we diagnose this? And this is on page 107. You're going to use culture and staining techniques using specific chemical specimens and also drug sensitivity tests. Your blood test can show variations in numbers of leukocytes. The leukocytosis would be present in a bacterial infection, where leukopenia would be in a viral infection. You can do a differential count, a C-reactive protein test, or an ESR, which is an erythrocyte sedimentation rate. And these are all different blood tests to try and diagnose these infections. You can also have immunological testing of bodily fluids, such as antigen identification or an antibody titer to see, which antibody at what level would kill off what you're looking for to, kill off the bacteria or the virus, whatever, the bacteria. So what about guidelines for drug therapy? Drugs should be administered and taken as directed, and this is actually a huge problem. This is one of the reasons that we have super bugs, right? Is because people start to feel better after, halfway through their antibiotic that they were prescribed, and so they stop taking it. And so then again, the fittest of the fit survive, right? And so those are the ones that are going to continue to reproduce and therefore cause even worse infections. So make sure that if you're prescribed 14 days of antibiotics, take 14 days of antibiotics. Also, antimicrobial drugs should be taken until they're prescribed. Again, like I said, used or until a new drug is prescribed. If symptoms continue without reduction, make sure that you either contact your pharmacist or your doctor so that they can potentially prescribe a different medication. Do not use drugs prescribed to other clients or other infections because they're specifically made for that one, not made for, but they're targeted for that one infection that you're working on eradicating. And then if the drug resistance is known to occur with the infection, then multi-drug therapies may be necessary. Classification of antimicrobials, we have antibiotics, and these are drugs that are derived from organisms, and they can also be made synthetically. You have antimicrobial, and these include antibacterial, antiviral, and antifungals. You have bactericidals, and these are drugs that destroy bacteria. And then you have bacteriostatic, and these are going to be inhibiting the reproduction of bacteria. Broad spectrum antibiotics, these are effective against both gram-positive and gram-negative organisms, hence the broad spectrum. The narrow spectrum are going to be effective on either or, so either gram-positive or gram-negative, so it's more targeted and specific. You also have first and second generation drugs. The first generation, those would be your original drug class, where your second generation would be like a later version, which may be more effective and may be more tolerable, or it may be more easily administered. So like, for instance, penicillin was one of the first ones, and then like amoxicillin would be like a second generation or things like that. So they can improve on it and target it a little bit better because pharmacology continues to develop amazing new drugs. So what about the mode of action of these different antibiotics? Well, they can interfere with bacterial cell wall synthesis. An example of that would be penicillin. This is kind of like the first figure that we just showed. You can also increase the permeability of the bacterial cell membrane. An example of this would be polymyxin. You can interfere with protein synthesis. An example of that would be tetracycline. You can interfere with the synthesis or the making of essential metabolites. And an example of these drugs would be sulfonamides. Or you can inhibit the synthesis or the making of nucleic acids. An example of this would be like ciprofloxacin. What about mode of action? Well, drugs may act by either blocking entry into the host cell, inhibiting the gene expression, or inhibiting the assembly of the virus. Again, this is exactly like what I showed in that first diagram when we first started this. Antifungal agents, these may interfere with mitosis in the fungi, and they may increase fungal membrane permeability. Most antifungal agents are administered topically, so to the actual location, to the skin or the mucous membrane. And fungi are eukaryotic cells, and the antifungal agents are therefore often toxic to the animal and human cells, and treatment therefore requires strict medical supervision. And then the final slide is antiprotozoal agents. And these are similar characteristics to the antifungal agents. The protozoans are going to be eukaryotic cells, and so we could damage normal body cells because remember, our body cells are eukaryotic cells as well. Many pathogenic protozoa have several stages in their life cycle, so this therefore requires treatment with different agents at different stages of the cycle to actually target that protozoa, but not our body cells. All right, and that wraps it up for chapter 6.